Review questions. Select the one best answer. 1. A double-stranded RNA genome isolated from a virus in the stool of a child with gastroenteritis was found to contain 15% uracil. What is the percentage of guanine in this genome? A 15B, 25C, 35D, 75E, 85. Answer explanation. The correct answer is C, 35. For double-stranded nucleic acids, Chargaff's rules apply. In double-stranded RNA, uracil pairs with adenine, and guanine pairs with cytosine. Therefore, the percentage of U must equal the percentage of A, and the percentage of G must equal the percentage of C. Given that uracil is 15%, then adenine must also be 15% due to complementary base pairing. The total percentage of A plus U is 15% plus 15% equals 30%. Since the total percentage of all bases in a nucleic acid molecule is 100%, the remaining percentage must be accounted for by G plus C. 100% to 30% equals 70%. Because G equals C in dsRNA, we divide 70% by 2, 70%/2 equals 35%. Therefore, the percentage of guanine in this dsRNA genome is 35%. 2. What is the structure indicated below? Description of the structure: a molecule showing an adenine base linked to a ribose sugar, and no phosphate group attached. The carbons of the sugar are numbered 1 foot to 5. A purine nucleotide B, purine C, pyrimidine nucleoside D, purine nucleoside E, deoxyadenosine. Answer explanation. The correct answer is D, purine nucleoside. Let's break down the structure presented. The nitrogenous base shown is adenine, which is a purine due to its double ring chemical structure. The sugar component is identified as ribose because it possesses hydroxyl groups at both the 2 foot and 3 carbon positions. If it were deoxyribose, the 2 foot carbon would have only a hydrogen atom. A molecule consisting of a nitrogenous base covalently linked to a 5 carbon sugar, but without any phosphate groups attached, is defined as a nucleoside. Combining these observations, the structure is a purine linked to a sugar, making it a purine nucleoside. Specifically, this molecule is adenosine. Option A, purine nucleotide, would require at least one phosphate group attached to the 5 foot carbon. Option B, purine, refers only to the base itself, not the base sugar complex. Option C, pyrimidine nucleoside, is incorrect because adenine is a purine, not a pyrimidine. Option E, deoxyadenosine, would imply deoxyribose as the sugar, which is not consistent with the presence of the 2 foot OH group. 3. Endonuclease activation and chromatin fragmentation are characteristic features of eukaryotic cell death by apoptosis. Which of the following chromosome structures would most likely be degraded first in an apoptotic cell? A bar body B, a 10 nanometers fiber C, 30 nanometers fiber D, centromere E, heterochromatin. Answer explanation. The correct answer is B, 10 nanometers fiber. Apoptosis, or programmed cell death, involves the activation of specific endonucleases that cleave DNA. These endonucleases preferentially attack the most accessible and open regions of DNA within the chromatin structure. The 10 nanometers fiber represents the beads on a string structure, where DNA is wound around histone octamers to form nucleosomes, with stretches of linker DNA connecting adjacent nucleosomes. The linker DNA regions, which are not protected by histones, are the most exposed and therefore the most sensitive regions to nuclease attack. This leads to the characteristic laddering pattern of DNA fragments observed during apoptosis. The 30 nanometers fiber is a more condensed structure formed by the coiling of 10 nanometers fibers with the help of histone H1, making the DNA less accessible to enzymes. Heterochromatin is a highly condensed and transcriptionally inactive form of chromatin. Its tight packaging makes it largely resistant to early nuclease digestion during apoptosis. Therefore, the unprotected linker DNA within the 10 nanometers fiber would be the primary target for endonucleases during the initial stages of apoptosis, leading to its degradation first. For a medical student working in a molecular biology laboratory is asked by her mentor to determine the base composition of an unlabeled nucleic acid sample left behind by a former research technologist. The results of her analysis show 10% adenine, 40% cytosine, 30% thymine, and 20% guanine. What is the most likely source of the nucleic acid in this sample? A. Bacterial chromosome B. Bacterial plasmid C. Mitochondrial chromosome D. A nuclear chromosome E. A viral genome. Answer explanation. The correct answer is E. Viral genome. The key to solving this problem lies in applying Chargaff's rules. For any double-stranded DNA, the percentage of adenine must equal the percentage of thymine, and the percentage of guanine must equal the percentage of cytosine. Let's examine the provided base composition. Adenine equals 10%. Thymine equals 30%. Cytosine equals 40%. Guanine equals 20%. Comparing the percentages, we see that A does not equal T, and G does not equal C. This significant deviation from Chargaff's rules is a strong indicator that the nucleic acid molecule is single-stranded, not double-stranded. In single-stranded nucleic acids, there is no complementary base pairing across strands to enforce these ratios, allowing for highly variable base compositions. Now, let's evaluate the options. A bacterial chromosome, typically double-stranded circular DNA. B. Bacterial plasmid, double-stranded circular DNA. C. Mitochondrial chromosome, double-stranded circular DNA. D. Nuclear chromosome, double-stranded linear DNA. All options A, B, and D represent double-stranded DNA molecules, which would adhere to Chargaff's rules. E. Viral genome. While many viruses have dsDNA or dsRNA genomes, some viruses, such as parvoviruses, possess single-stranded DNA genomes, and many RNA viruses have ssRNA genomes. In a single-stranded molecule, the base composition is not constrained by complementary pairing, and therefore, it will not necessarily follow Chargaff's rules. Therefore, a viral genome with single-stranded nucleic acid is the most likely source for this sample.